Allele exchange or gene replacement in E. coli has been around for a long time. The illustration describes gene replacement in Pseudomonas, but the concept is the same for E. coli. A suicide vector that is unable to replicate is constructed to contain a mutant gene replacement. Under selection, this suicide vector undergoes the first crossover event and becomes incorporated into the E. coli genome. A counterselection step, in this case sucrose selection because of SAC-B on the integrated plasmid, causes a second crossover event to occur that removes the plasmid from the chromosome. Depending on where the second crossover event occurs, the end result will be a reversion back to wild-type sequence or the wanted mutation. Now, there are advantages. This does allow for precise, unscared, and unmarked mutations. However, there are some disadvantages. Depends on host cell recombination systems, requires mutant gene replacement construction on a plasmid, counterselection compounds may be toxic, and target strains may require certain mutations.